So, okay. So thank you very much, and thank you very much to Betsy for inviting me. This has just been a great couple of days and a really great symposium. I've had a great time. So thank you very much. And to change gears a little bit, I think the best way to tell this story and the molecular epidemiology and how we integrated genomics in the hospital environment is just to sort of tell it from a personal standpoint. So I'm an ID physician, and we had a clinical problem, and then I think in my heart I'm an evolutionary biologist and or wannabe, and molecular epidemiologist. And so with that in mind, I always like to start with a case. So just to orient you to this case, it kind of falls on the heels of the Staph aureus talk that Dr. Popovich gave. So 27-year-old female, admitted to our hospital with a history of chronic pain, has a bacteremia with Staph aureus. Because we couldn't get IV access, she had to have a central line placed. That's a catheter that goes into an arm because the peripheral veins weren't accessible. 6 days into that, she has a gram-negative rod grow from her blood. And it looks like this. So this is an Enterobacter cloacae that is highly resistant. As you can see, you don't need to know much about microbiology, but everything there is pretty much listed as resistant. Susceptible only to amikacin, which is a really lousy drug. And so, you know, what's unusual about this case is most of the patients that get KPC at UVA, at our institution, are there for quite some time. They often have a lot of comorbidities and they're very sick in the ICU. So this was a pretty unusual case in that she was from the community. So how in the world could this case involve the hospital environment? Well, in our work to try to figure out where our reservoir of KPC was, we had sampled the sink that patient, the room of the sink where that patient was admitted 2 months prior, and had found a KPC producing Enterobacter cloacae in the sink drain. So we did long read sequencing, and I'm going to talk a lot about different genetic elements today, and hopefully it's not too boring, and those who enjoy thinking about that will get something out of it, and those who don't, it won't be too painful. But basically, just a little cartoon depiction of the Enterobacter cloacae from the drain, isolated in January, and the Enterobacter cloacae from the patient, isolated in March. Essentially, the chromosomes were identical with 3 SNP differences. We did long-read sequencing to resolve the plasmids. Both isolates had 2 KPC harboring plasmids, which is a gene of carbapenemase resistance. And the only difference was there was an additional insert in the environmental isolate of 14 kb, 12 here and 1 there. So fairly compelling. What we then also learned about this patient is that she was an IV drug abuser and was actually using water or wastewater in her room for recreational injection into her line, likely. So how did we even get to that point? And how often is this happening? What I also don't want to do is overblow that UVA is the only place this is happening. We've just been looking really hard to figure out a problem and to figure out multi-species acquisition. And so what's going on in the rest of the United States? So carbapenem-resistant Enterobacter acae were basically unheard of in 2000. Really, there wasn't much dissemination in the US before 2010, and now almost every state has had at least 1 or more carbapenem-resistant Enterobacter acae. The reason these are so problematic is because carbapenems were thought to be the last line agent against enteric bacteria. Enteric bacteria, like E. coli and Klebsiella, cause a lot of human infection. And so these are really precious drugs. When you look at carbapenem-resistant and carbapenem-susceptible Klebpneumo, there are worse outcomes likely because of the lack of available agents. Not to overblow this, we do have several new agents coming or available to use against these types of organisms that are anti-KPC. So things are going to get better, but preventing the spread is critical because these organisms blow through these drugs fairly quickly, just as a beta-lactamase aficionado. So KPC, what is it? So I tracked a gene. I didn't realize that's what I was doing, but most outbreaks are MRS, a strain. You're tracking a strain, or you're tracking a species, or you're tracking a clone. But I was interested in the in the way that KPC was moving between species. And we also had an opportunity to look at that because that's what we were seeing in our hospital. So to talk about the gene that I tracked, some of the reason that we could track it is because it has such a dramatic phenotype. When you knock out, when you have KPC, that means the organism is pretty much not susceptible to penicillins, cephalosporins, aztreonams, and carbapenems. And as a clinician, these are what you treat these types of organisms with. It is, importantly, on this mobile piece of DNA in almost all the descriptions. It's a 10 kb transposon, and for those who like transposon biology, it's a healthy transposon. It's got a resolvase, transposase, and it can freely move and does frequently. KPC gene around the globe, there have been several places. This has been the most successful carbapenemase gene, and it first arose in America, went from New York to Israel, also the Caribbean early, and China, which also sort of speaks to some of the geopolitical movement out of New York probably, and then South America, and then hit Italy and Greece fairly hard. It's now the most predominant in the UK as well. But what's interesting, oh, sorry, I goofed up. Uh-oh, sorry. Let me do that. Oh. All right. Sorry. Oh, there we go. Okay. So, sorry about that. So, the thing about KPC, the gene, it's most typically in almost all outbreaks and regions that it's taken off, it is seen in a Klebsiella backbone. And Dr. Bachman gave a really nice talk about Klebsiella pneumonia. So this is a really important and relevant hospital-acquired organism. And so in most places, it was the KPC gene has been paired with this very successful clone, ST258. And so I just, by way of background, it's important to understand that because that's relevant to what we saw. So, 2007, in August, I was an infectious disease fellow. We had a patient who was on the transplant service, and he was transferred from a hospital in Philadelphia with Klebpneumo and Kleboxytoca. And after 30 days, these popped up in his blood and in abdominal, and they were not susceptible to carbapenems. He basically was in our hospital for 4 months before succumbing to the infection. One of the things we were instantly like, oh, we'll just do a case report because there hadn't been at that point very much movement of the gene between one species to another. And so we went ahead and took the Kleboxytoca and the Klebpneumo, 2 different species, and through southern blotting and old techniques, we basically figured out that there were 2 different plasmids. One was the Kleboxytoca was much larger. And so we had 2 plasmids introduced to our hospital. No big deal, case report to follow. But the issue for us was, here is our index case, and this is our early epidemiologic curve of KPC in our hospital. If you look at this and you're an epidemiologist, you can say, yes, this looks like an outbreak. You have an introduction and then persistent spread. And we're confident there wasn't KPC in our hospital before that. And so here's the Kleboxytoca, here's the Klebpneumo. The next patient that popped up with one was a Klebpneumo, but then an Enterobacter, and then an E. coli, and a Citrobacter, and Enterobacter absuriae, and so on. And so there was a lot of what we thought maybe was a gene moving in between species. So, just to orient those who are not familiar with gene movement between bacteria, here's sort of what had been happening in the rest of the country, a clonal outbreak, a very successful strain of Klebpneumo spreading from patient to patient in hospitals and within regions. Again, KPC is contained within that 10 kb transposon. Here's the gene depicted. Here's the plasmid. These are not to scale. This is the 5 megabase chromosome. And so this is what was spreading for most of the country. But what we thought we were seeing was this gene moving to a new strain, moving to a new strain, new species, new strain. And so we thought we had a plasmid-mediated outbreak. What we also weren't 100% sure on, but was, can this transposon be moving between plasmids and moving also? So we developed a PCR that tracked both PUVA01 specific to the region outside of the 10 kb transposon for PUVA01 and PUVA02 and applied it to all those different species. And we found, yep, look at that. We've got a plasmid-mediated outbreak. So 80% of our isolates had PUVA01, 15% PUVA02, and so on. What's also important to note during this time, so while we're having all this transmission, you're like, well, what are you doing to stop it? So we were trying to figure out why were we seeing this transmission, and we had assumed, and based on literature, it looked like silent reservoirs were probably going to be patients who were colonized that didn't have enough of an organism to have an infection. And so we developed perirectal screening, following the CDC guidance, and then sort of tweaked it for our own microlab and outbreak to look across all species for silent colonization via the perirectum. So swabs go into a broth with an ertapenem selection. We put it on a KPC chrome agar. Enterobacteriaceae that are carbapenem resistant are shown pigmented in here, and then we do a phenotypic test, and then I was taking all these into the research lab and doing PCR to confirm KPC. So the first time we did perirectal colonization, a perirectal colonization sweep in our surgical ICU, in fact, 12 of 14 patients were colonized with KPC. So we had found the reservoir. It was silently colonized patients. We immediately changed a lot of infection control practices. We did a lot of the things, and I won't go into the details. They're very akin to what Kyle had described, Dr. Popovich had nicely described of enhanced hand hygiene, contact precautions, cohorting patients, deep cleaning. And it worked. We had a decrease in transmission. We also had multi-drug resistant Acinetobacter introduced and did not see prolonged transmission. Except that we kept seeing these low-level, infrequent acquisitions of new KPC, even with all of that enhanced screening. And we do roughly 9,000 perirectal screens a year at this point. We screen many admissions, we screen, and I can go into the details of that, but we do a lot of perirectal screening. So the group at Oxford, Derek Crook's group at Oxford, saw this work and said, hey, we think we can solve that plasmid-mediated outbreak in full with Illumina short-read sequencing. Are you in? I was like, yeah, of course I'm in. I would love to do that. So we took the first 5 years of transmission of isolates from 2007 to 2012. It was 281 isolates that we Illumina sequenced, and I'm just going to, just for entertainment reasons, walk through briefly how we did the initial plasmid workup, and then look at Club Pneumo, because I think there's interest, and then go into sort of the rest of the outbreak. So, sorry guys, did it again. Is that working? No. Did that work? Okay. So, what we did was, we electroporated, so extracted the plasmid from the index case because we were postulating that index plasmid, PUVA01 and 02, were seen throughout the outbreak. So we took both of those isolates, extracted the plasmid DNA, electroporated it into a known E. coli background, Illumina sequenced it, got all these short reads depicted in cartoon here. You subtract the E. coli genome, and we de novo assembled the plasmid. Easy enough, and it worked. So PUVA01 in detail, shown here, is a 43 kb plasmid that, here's the transposon shown here. The KPC gene is in red. And a lot of the machinery on this plasmid is devoted to conjugation and plasmid maintenance. When we blasted about 30 kb of this into NCBI, there was nothing in the database that looked anything. There's just no hits, no matches for this DNA. What I'll say now is this is a paper from the end of 2016 from Barry Kreiswerth's group that looked across all Enterobacter cloacae in the US from whole genome sequencing. And you can see Michigan has PUVA01, unfortunately, and so do a lot of other states. So this plasmid has legs and has gone elsewhere, or was elsewhere, and we're now finding it. And then here's just the larger PUVA02 kleboxytoca plasmid. And again, what I'll draw the environmental microbiologist attention to is there is a lot of heavy metal resistance on this plasmid, which is, somewhat unusual if you just think about why are humans carrying different things. So here was the club pneumo. So we first took a look at club pneumo partly because we felt like that was going to be the easiest thing to do in terms of what was the available genomics at the time and how we could do it. So you've already been oriented to sort of phylogenetic trees. This is a really large phylogenetic tree, so 16,000 SNP differences. These are actually other species we now know, but this is what we published. There are imported strains here depicted in red. So that means the patient was either identified as having a carbapenem-resistant club pneumo before transfer to our hospital and had never been admitted to our hospital, or they were identified within 48 hours. And what you see is this clustering of ST258 that was being seen elsewhere. And so it was like we were an island of unique acquisition in a sea of ST258. And here's our index case. But what you also see is depicted in blue, patients who were in our hospital for more than 48 hours and had overlap on a unit with another patient with KPC, with a KPC producing isolate. We did not enforce that it had to be the same strain, nor even same species. And we found that, yeah, there were a lot of locally acquired. The indeterminates mean that we're not sure where they got it because they were in our hospital for a while before we identified it. So then we took that de novo assembly, and here is depicted, we blasted back in, did it have PUVA01 or PUVA02? And we found, yep, it looks like it does, especially in this clade here, ST941. And so what this is a time-stamped phylogenetic tree of sort of the evolution. And so it looks like both PUVA01 and PUVA02 went into this ST258 strain of club pneumo, which seems fairly successful in our hospital, at least twice. If you believe this, it looks like it went in at least 3 times, that PUVA02 went in a separate time. However, this data should be taken with a grain of salt, and I'll tell you why in a moment. So then what was going on with the rest of the species? So this was work done, led by Anna Shepherd at University of Oxford, and this is too hard to see, but just to orient you to, these are a lot of trees. Enterobacter cloacae, club pneumo, club oxytoca, Citrobacter frundii, there's all those imported patients. But we did the same thing where we mapped back in on this really diverse outbreak, patients who are the PUVA01 isolate, and you see that a lot of these strains seem to be carrying that plasmid. So when we looked across, here's sort of the numbers. 66% of unique patients actually had PUVA01, 59% of the strains of the 281 that we sequenced, 20% roughly had PUVA02, and then 24% had neither. And again, those were enriched for those club pneumo coming in from somewhere else. So it's fairly compelling for a plasmid-mediated outbreak, but we needed to do long-read sequencing to make sure that we weren't being fooled about were those plasmids still carrying the KPC gene. And it's in the weeds of short-read sequencing that I can tell you that you can't resolve this, and I can go into detail about why, but ultimately you need to do long-read sequence to say for sure if the KPC gene is still associated with the plasmid on which, even though both are in isolates. And so here is the long-read sequence data. We randomly selected 17 isolates, and I have great regrets about randomly selecting isolates because we didn't select very well across a bunch of clades, and so we have a lot of redundant data, but it was the pure thing to do, and it was my colleagues at Oxford that felt it was very important to randomly select, so that was very good, because it is really random. We had a lot of these isolates were predicted to have PUVA01 or PUVA02 by our mapping methods. But when you look at what plasmids were actually carrying the KPC gene, it was a minority of them. And so we had been sort of fooled. Yes, PUVA01, to be clear, that plasmid DNA is in those isolates, but KPC is no longer associated with it. And so the other thing to note is a lot of these strains have a lot of plasmids, and is not for the faint of heart. So here's the structural diversity of PUVA01 across all those We had 11 isolates with PUVA01. A lot of the time, more than almost half of the time, KPC was no longer associated with it. It had done a lot of interesting things, including this demonstration of a co-integrate structure where it looks like the transposon was trying to move into another plasmid and did not completely resolve. So it's like hooked to another plasmid, but so KPC is in that plasmid twice. A lot is going on here molecularly within these isolates. But what was also really important was that the TN4401 mobile transposon had inserted into another transposon, TN2. And this is the transposon that is famous. It's one of the original transposon and famously carries a TEM gene, which is a different beta-lactamase. And this is littered throughout Enterobacter ACA, especially E. coli. And so this gives rise to an opportunity for this transposon to move very frequently through homologous recombination. And so when we had done our little plasmid PCR, we were actually looking within this TN2 region and not out here. And so that's part of how we got fooled into thinking, oh yes, these are all the same plasmid. They're all, we even did long-reach PCR to resolve this, and we were duped. So there were 11 different KPC plasmids over those 17 unique isolates. So what we've sort of put together is this issue of the concept being sort of a Russian doll, that there's a carbapenemase gene sitting in a mobile transposon, which we have evidence has moved independently, sitting within another transposon, which we know has moved multiple times through homologous recombination, sitting in a mobile plasmid that we know has moved between strains and species. And so it is within a single hospital, within a single outbreak, we've been able to see this because KPC is not very frequent in the community yet, because we were looking really hard for silent colonization, and we were good at keeping Kleb pneumo from transmitting in our hospital. I think if we had a Kleb pneumo outbreak, that's what we would have paid attention to, but we were able to sort of keep those down, and so we had this sort of unique multi-species opportunity that arose to look at this. And I love this paper, and this is a word map of frequency, and this is work from 2010 where they looked at all protein coding sequences across archaea, bacteria, eukaryotes, and viruses. And the most frequent protein coding sequence is transposases. And so if you think that we are going to beat this evolution, I think that we are not. This P150 is also another mobile element. It's not what I would have predicted as sort of energy generating protein coding sequences. It's transposases. Organisms are fit to evolve. So from all of that work, could we make any sense of the transmission, or how could we apply this to our transmission within our hospital? So this is a depiction of an idealized sort of outbreak where you'd have the introduction of a Kleb pneumo, and then you'd have the next patient. And in solid circles, you'd have those two patients were on the ward at the same time. And so that patient carried KPC. Somebody forgot to wash their hands or clean the equipment, and it went to the next patient, and the next patient, and the next patient, and so on. Occasionally, you might see these hollow circles where this person got KPC Kleb pneumo, and you don't know who they got it from. But this is a pretty, standard idealized clonal outbreak. This is a depiction of our outbreak. And so here, shown on this axis, is the 62 different unique strains separated by more than 500 SNPs. Different species are depicted in different colors. Imports are depicted in X, and you can see all the ST258s on that axis. And solid circles are patients where they were on the ward at the same time as another patient who got that strain. And so you can see here there have been times where we've had clonal transmission. This is when we did our first perirectal screening with Klebsiella and Enterobacter cloacae. But what we have a lot of is these hollow circles where the patients are seemingly getting it from you don't know where. They have no ward overlap with another patient that carries that strain. And what's more bothersome is these huge gaps in time where you see this strain and then it doesn't appear for another 2 years. Where is that strain in the interim? And so patient-to-patient overlap or transmission only accounted for 30% of our outbreak. And then we started seeing KPC erymonis. And so anybody who knows microbiology is going to say erymonis is a water bug, and that's what I said. And we freaked out and started testing all of our municipal water and found nothing. CDC was involved, VDH was involved. We did this 2 times through and could find nothing. So we let it go. Just to let you know sort of what our erymonis patients looked like. They were, this is what our typical KPC patients look like. They are not the worried well, in that they are in the hospital for a very long time before they get an organism. And a lot of these patients had more than one species. And a lot of the patients expired, although very few expired from a KPC infection, just shown here. And so what we started wondering about was the water, and it was the wastewater. And, everybody in this room is kind of keyed into this, but there were a lot of people in the field, and still sort of are, that are not sure this is how relevant this is. And anyway, we can talk about that after. But so the traditional idea is that a patient's colonized with a strain, and this is probably true of a lot of strains and species. And so it's probably very unique and nuanced as to how transmission occurs. But Dr. X touches patient colonized, doesn't wash his hands, and then goes and diddles with patient B. And so transmits through poor hand hygiene. What I think is we started wondering about the sink trap reservoir as potentially a source for transmission. And so I just want to make sure that people are very clear about what I'm talking about. When we are not talking about this incoming water. We are talking about everything else I'm going to say is sort of from the wastewater side out. And so from the drain, just to orient you if you're not a plumber or environmental microbiologist and haven't thought about drains in a while, there's standing water in this drain. It keeps sewer gas smell from off-gassing. That's the only intent of this standing water that's full of nutrients and antibiotics sometimes. And it's about 10 to 4 inches from this drain piece, and then the sanitary stack, which connects to all the other wastewater, depending on how your hospital's built. What I would say is that we might be looking at an issue that we can see more clearly now that there's higher risk genes of drug resistance. And so because carbapenem resistance genes are so noticeable clinically, we might be seeing more of this. There are 70 reports as of the beginning of October that depict outbreaks or implicate sinks and sink drains in transmission of nosocomial organisms. 21 of those have been in 2017. And so this is being picked up, and this is being increasingly recognized as an issue. And I think it's because we have more resistance and this gene exchange that can occur. I would also say that we don't completely understand nosocomial transmission of ESBL, and never have. And that when you do things like you do for Kyle's study, where you implement contact precautions, it doesn't usually work very well for other beta-lactamase producing Enterobacter ACA. So maybe this has been happening the whole time, and we just haven't recognized it. The first report was in 1969, so from Mass General, and they put phenol down the drain. So if you start thinking about the wastewater, and this is probably a lot of people in this room may know this, but it's like the perfect place for organisms to develop and evolve drug resistance. You seed a sink with a patient's isolate, so you put genes of drug resistance in there, you give it lots of water, which is really good for life. You add selective pressure through antibiotics that are used in the hospital. You dispose of nutrients. In the U.S., at least, we tend to think of sinks as garbage disposals, and we throw a lot of waste down the sink drain and without thinking about it. If you have a half-finished cup of milk in a patient room, it often goes down the drain, and then you can't clean that area because there's a drain on top of it. So when we looked in our surgical ICU at UVA for KPC-producing bacteria, so when it occurred to me, I was just coming back from Oxford after sabbatical, and I realized this was probably happening, and I just went in and started sampling, not really talking to anybody about it. And I think if I had been a hospital epidemiologist, I may not have ever done that. I just had to know if our drains were colonized. And so, and it was slightly, you'll see what happened. So basically, 14 of 15 patient care sinks were colonized with at least one species of KPC-producing bacteria. At that time, this was a picture taken on that day. There's this counter space that's next to the sink, and this is not supposed to happen, but we give our nurses no horizontal space to work in the ICU room, so there's nowhere to put patient care equipment. So it was all piled up around the sink. So, what's going on with the microbiology? So back to that cool microbiology of sinks, which I think will be appealing to this audience, although terrifying that that's what we found. So we tried to figure out, I had postulated that what you would see is a unique ecology in each sink trap. It gets seeded at some point, and then that sort of develops and flourishes over time, and all the genes get exchanged within that sink trap, and that's not really what we found. Here's, again, long-read sequencing depicted. We have a Rowatella with a KPC gene here, the transposon. It's in that TN2 transposon, and it's carrying the KPC3 allele. There's also another transposon inserted into this transposon. So, and it's all sitting on a ink-N plasmid backbone. Very different from what we see in the Citrobacter frundii that's in the same sink drain at the same time. It's on the PUVA01 isolate or plasmid that I had told you about. And then also there's this ink-LM 86 kb plasmid backbone. And what's really interesting is that really long plasmid co-integrate structure is now resolved. And so there's pretty compelling evidence that these are evolving and resolving. This was from a patient in 2012. We didn't look in the sink until December of 2013. And so, yeah. So again, testing my hypothesis, are these isolates all the same are they related within a sink, or are they same across the sink? And what we find, this is work of Nicole Stosa, and she presented this at the MAC-EPID conference. And again, orienting you to, here's the layout of the SICU, a phylogenetic tree. The branches on this tree are exceedingly short. This is 5 SNP differences across. And here, they all have the same TN4401 variant. They all have the same allele, and they have the same target site duplication, meaning the transposons likely inserted into the same place. So all highly related across all these different sinks. So there seemed to be a flora across these sinks rather than a unique isolate. We saw the same with Enterobacter, cloacae. Across the sink traps, there was relationships. These are all fairly highly related. These two branches are a little bit distinct, but they all carried the same mobile element. And the same with Aeromonas. So more similarity across sink traps than within. So, again, that's great, Amy. Interesting biology. But what were you going to do? So we went to the literature. I called everybody that I could get a hold of that had had one of these outbreaks and described it and asked them what they do, what should we do. And this is a review that we put together to try to figure out what the most effective intervention is. And it's kind of a busy slide, but ultimately shown here in gray. So to summarize, this is 31 different analyses or different papers that have been published about sink-related outbreaks. Some of them in municipal water. It was all carbapenem-resistant organisms, so not just Enterobacter ACA, but we looked at Pseudomonas, Acinetobacter, and Enterobacter ACA. And this work was led by Alice Kinsey-Gordon and Nicole Stosa, and we all worked on it. But and what we found was that the gray were things that were done that were not effective. And what I would just draw your eyes to is there's a lot of gray on this graph. So a lot of things that people have tried have not been very effective. When you break out just Enterobacter ACA, all of them were associated with drains or sinks, no association with faucets. And what's interesting, again, is a lot of gray, but we thought bleach kind of looked promising and maybe replacement of the plumbing altogether, and some hydrogen peroxide maybe has some temporary effect. And so that's what we decided to do. So we did a whole ICU overhaul, get everything off of the sink. We worked with nursing and facilities to redesign our whole ICU workflow so the nurses didn't have to put anything on the sink. We put up placards here. We still audit whether or not people are putting stuff next to the sink. And then we removed all the drains from the drain to the wall and said, well, let's keep it from coming back. We'll pour bleach, hydrogen peroxide, or ozone-impregnated water down the sink once a week. So what we didn't enforce is we'd shut down the ICU for 9 weeks to figure all this out. And so when we moved patients back in, we didn't put up or couldn't put up a rule about not having patients who were colonized with KPC coming back into the unit. And so we had a patient that was transferred back into a room with a brand new trap who had carried a Serratia marcescens and a Kleb pneumo. And just doing the long read sequencing there, you can see that the Serratia and the Kleb pneumo depicted in cartoons have completely different KPC plasmids. So they're not sharing plasmids. He goes back into so those isolates were identified in November 2013. In February of 2014, he goes back into that room, and this is the isolate that we find in his sink trap. The Kleb pneumo has now picked up that Serratia isolate, and the transposon has moved between plasmids in a replicative fashion. What I would say about that bacteria, and I think this is one of the most important things, is something is really wrong with this bacteria. Bacteria should not do this. Healthy bacteria should not be picking up stray DNA. It's a huge stress on bacteria to do that. And I think figuring some of this out is figuring out what's wrong with this bacteria. And there's a lot of, we can talk about that after too, what I think is wrong with that bacteria. And so you can see here it moved over, and there's even more to this story, but for brevity. So, okay. And again, just to orient you, we thought we had a plan. We'd remove all the traps. We'd put back. In 10 days' time, 5 of 15 of those patient sink traps were back colonized with KPC-producing bacteria. And so, but we were like, let's soldier on and just keep pouring bleach down 3, hydrogen peroxide down 3, ozone-impregnated water down 3. We'll do this weekly. We'll have 3 controls, and we'll see what works best. And then a month into that, we started seeing Serratia, KPC-producing Serratia in high numbers. And so, again, for the clinicians in the room, this is a 31-year-old, chronic liver failure, GI bleed. He was doing better. He basically gets this. So Serratia marcescens is really intrinsically resistant to antibiotics at baseline. So then you put in a KPC gene, and it's very, very difficult to treat. Things like colistin are intrinsically resistant. I personally think tigecycline is probably intrinsically resistant. And so it was bad. So this is 30 days after we did that intervention with the bleach and the hydrogen peroxide and the ozone. And also, because we knew our medical ICU had the same problem, although we didn't change out the traps, we started pouring bleach and hydrogen peroxide and ozone-impregnated water down those because we didn't want to not do something. So we were doing this in 2 units simultaneously, and we started seeing KPC Serratia in our patients. So all of these are depicted as patient isolates. These are all environmental. This tree is really, really small. So this is 1 SNP difference. These are all the exact same clone. There's no more than 10 SNP differences across this tree. Oh, did I do it again? Oh, sorry, guys. So, from a molecular epidemiology standpoint, I was like, oh, we finally have a clonal outbreak. And so we can make some sense of it. But here, depicted here, is all the Serratia that was acquired by patients, so 37 patients, 32 of whom acquired after we did the bleach hydrogen peroxide. They all have the completely same plasmid, same TN4401 structure. It is not in the TN2 element, and they're all just pretty much identical. What I would say from a molecular epidemiology standpoint, this is all of our Serratia. This has not been published. It's a tiny tree. But you would think maybe we could start making inferences about transmission because we have a clone. But that's not working either because, as you see here, you have this nice case where you've got a patient that goes into this room, 5133, on this date, and then essentially the environment's positive a month later, and then a patient goes through that room again, and acquires that same isolate with no SNP differences. Fairly compelling story. However, down here, you see that the drain swab is in this room, and none of these patients have ever gone through that room. And so you're just struggling with how are you going to make sense? And when you have so little genetic variation, it's all essentially the same. And, you know, people who want to get into the weeds of, again, evolutionary clocks, Serratia has a very slow evolutionary clock. And so there's just so little genetic change, you can't make transmission inferences very easily. So the hospital was like, Amy, out of the hospital, we'll build you a sync lab and figure out what the fix is for this problem, and then you come back and implement it. And so we worked for about a year trying to figure that out in a sync lab that we erected, and then Shireen Cote, environmental microbiologist, joined our group and helped finish this work. And so we were trying to figure out transmission, and as you can imagine, the municipal water, how do you tell the municipal water, which is not sterile, from the wastewater, and how are you going to put an indicator? And I had tried to reproduce a lot of people's work around this, and I couldn't. And so that's what prompted us to come up with this GFP E. coli. And so what we did was we put GFP E. coli essentially into the P-trap water, and nothing happened. When you hit that with water, nothing happens. If it's just confined to this area, it stays confined to that area. In fact, when the water runs, it flushes down. But what happens is if you add nutrients to this system, this E. coli grows up at about 1 inch a day to the drain. And as soon as GFP E. coli is sitting on this drain and water hits, it splatters all over the place. The other thing is that if you put GFP E. coli into this drain, you will see it show up in 28 days or less in this drain. And so these are all connected, and this is biofilm, and I'm not saying E. coli's swimming everywhere. It's connected, I believe, biofilm and water flow. And so here's sort of the dispersion maps of how we did this experiment, but up to 36 inches, we were seeing colonies of E. coli dispersing when water hit. So we were ready to sort of think about, okay, so we need an intervention targeting that drain, that drain plate, not the P-trap necessarily, but the drain plate. But before we go back into the hospital, we should just make sure there's not a reservoir that we've overlooked. And so enters the hoppers. So these things are toilet-like things that are in ICU patient rooms that we dispose of waste. They're behind a wall barrier, but they're actually open to patients, so you can kind of see the layout of the room. Not very well, actually, here, but here's the patient bed. Here's the hopper behind a barrier, but really open, and here's the sink. Oh, here's the bed, actually. And so we figured, well, we maybe better sample those, but I had actually thought that this was going to be a different situation because these seem cleanable. It seems like it's a porcelain surface, you could really clean it. It's not going to have a persistent biofilm, but I was wrong. 75% of our hoppers were positive for KPC-producing bacteria at baseline. 8 species, 30% of them have more than one species of KPC at any given time. 50% had Serratia, 25% Aeromonas. So we did an intervention where we put lids down on the hopper. And I know this seems easy and straightforward, and you would think that in a hospital there might be lids everywhere, but there's usually not lids on toilets in hospitals. We also, in our surgical ICU, to address the sink trap issue, we put these heaters on. So this is this German heating device. We had to get risk management involved because they don't have a CE mark or UL mark for use in the hospital in the US. But we were able to get these, and they heat to 75 to 85 degrees C continuously and have a vibration mode. So it's really hot. And they heat the P-trap water continuously. And so we did a study where we looked at all the patients who had acquired KPC before installing hopper covers and the sink intervention devices in a single unit. So the sink trap intervention. Those sink trap heaters cost $60,000 for 15 patient rooms. And so we did it just in one room. And then we looked at what happened. And what we found was before we had the interventions around the environment, we had 77, over 15 months, we had 77 KPC acquisitions, 33 clinical cultures, and 44 were just silent colonization. in the post-intervention period, sorry, the slide's a little goofy, we had 13 clinical cultures and 40 KPC acquisitions total. So across the hospital, this was completely statistically significant. So we finally had a drop in our KPC acquisition. And so you can kind of see here is what was going on before by month, and then our installation period, and then after. And this is a 60-day survival curve showing time to acquisition. It's not survival of patients, but time to acquisition, pre-intervention versus post-intervention. And so you can see that we had significance by log rank test. So in closing, there's a lot going on in the hospital environment. And I think that what I'd like to leave you with is part of the reason we saw this is just because I was just trying to figure out the biology. And I think we've come upon something potentially very important, although a lot of other people have seen this as important. I don't want to stand here and say, but I think in a way we've had this opportunity to see this because of this single gene marker. And we are going to understand more about biology and bacteriology with some of this, and then also the sequencing. I would also say that short read sequencing is not optimal, but I do think that some of the data exists within those isolates and within those genomes to make some sense of it. But we don't have the tools to do so yet. And I think, what are we going to do about hospital environments? I think is a very big open question, and I'm not sure. I'm not sure what to do. I actually usually end this talk just making sure nobody walks out of here thinking, you shouldn't wash your hands. You should wash your hands. Keeping in mind, just to put this in perspective, 77 patients in 15 months. We have I think in that time frame we had 16,000 admissions. So this is a very infrequent, very few infections, but they are serious when they happen. I don't want to overblow this, but I don't want to understate it. And so I think it's important that we don't know what to do, and I would caution about going after this. The last thing I'll say is part of the reason I haven't presented a lot of this data is I made an agreement with our hospital that I would not publish any of this data until we had shown an improvement, until we had some of a fix, because there's a lot of legal ramifications for the hospital, and I feel obligated because I'm looking so hard. It seems overblown. I think this is going on in a lot of hospitals in the United States. I'm not sure about that, but, people are sending me a lot of P-traps with carbapenemase genes in them at this point. So anyway, so thank you. Obviously, I didn't do this alone. There's a lot of people that did so much work on this, and so I'll just leave you with that. So any questions? Yeah, so I'm not sure, and I think there's so many interesting questions for that, and I think that's why we really need to be pairing with environmental microbiologists that have such a better handle on this, is that when I look at the DNA around those plasmids, there's a lot of heavy metal resistance. There's a lot of things that you wouldn't think would benefit a Enterobacter ACA in the gut of a human, and so it may be that we're co-selecting for resistance. What I would also say as a clinician, if you're treating somebody with cefepime for a ventilator-associated pneumonia, that's 6 grams of cefepime a day, and that's basically going out in the urine unchanged. And so that's just one room. That's a lot of cefepime, which is a very broad spectrum cephalosporin. And a lot of antibiotics go out in the urine unchanged. So I think it's antibiotics, it's things that we don't know and factors we don't know. What is the water? What is the hardness of the water? Is there something, a je ne sais quoi about our hospital and our water that helps promote this, or how does something like PUVA01 help promote it? What are the factors on there that are co-selecting? So ultimately we don't know. We are working with a lot of different agents to try to figure out some of that stuff in the sync lab. We now have 2 sync labs. So
yes, 2 quick questions. One, what was the protocol for the disinfectants going down the drain? Were they really high
Yeah.
Maybe you can mention that. I missed it.
No. So I haven't. So let's take showers first. So I don't know if you can see my mentality if I just sort of put blinders on. Our shower drains have KPC. We have not done a hospital-wide survey of shower drains, partly because we have our hands full and I'm not sure what to do. And a lot of our ICU patients who get these are not taking showers. So we've not had a problem. We actually don't have a burn unit anymore. We did see a fair number of these coming up in our burn unit, but we don't really have a shower associated with that. So we weren't seeing a lot of concordance between the patients who were able to take showers, like in the hem-onc ward, where patients are very vulnerable, do take showers, and transmission. We do see it, but we don't see it that frequently. So we have not focused on that, but just go ahead and then what we did was we took concentrated straight bleach, 50 cc's, and poured that into a P-trap, and the volume of the P-trap in our hospital is 120 cc's, so we figured we'd sort of a one-to-one, and then we'd sort of stand there and police it and make sure nobody used the sink for 5 minutes, and then we'd walk away. We wouldn't run it, so if it stayed there. Hydrogen peroxide, we used a 10% solution, and again, so it's going to dilute. And then the ozone, we had these ozone generating machines that we got, and I don't have a picture of it, but we would take and put this ozone impregnated water hose thing with a patient in the room because we were trying to, and see if we could, and then just run that for 5 minutes while we stood there and held it tight to the drain. We developed a sealant for it and a weight for it and the whole thing. And the reason we did ozone, and we were so keen on going through all that trouble of doing the ozone, we'd run it for 5 minutes, was because, and that was running at 20 parts per million. 2 parts per million for that study, for that part of it. And the reason we did that is because I just was thinking, if you start pouring all that bleach down the drain, I had huge concerns about the corrosion implications, and to be honest, just the environment of that much bleach load. So I was keen on ozone because it gets used up over time. It gets used up once there's no bioburden. It sort of, it just made more sense to me for that trap right there. The one thing about ozone is that for any physician in the room or whoever's done an asthma rat model, our infection control practitioner that was doing sometimes had asthma, and she would sometimes, so we had to, anyway, we don't do it anymore, but so that was our protocol. And then when we started seeing the serratia, we were like, okay, let's do it twice a week instead of once a week. And so we upped the.
Did you see one of those, one treatment was doing better than the others?
So, they did seem to have an effect while, and the shift to serratia was significant in that most of the other units where we didn't do this didn't have serratia as heavily in the drain as they did in the units where we did. But you did see a decrease. It was working to some degree, and there probably is a sweet spot for doing that. But I think that what I like to just sort of impart on is we're not going to win that war. I mean, we've got to figure out a way to get along with the bugs. And, we've heard this, Betsy say to Dr. Foxman. But I think that combat, has turned me off to it. But it might work. And if you had a one drain, and other people have had luck, like in Australia, they've removed a drain and now they do bleach. At the NIH, they remove drain and they don't use acetic acid, but they have a shower drain that's been persistently colonized in a custodial closet. And Tara Palmer was talking about it at ID Week. And they remove it, use a heavy cleaning solution. I forget what they use, but and that's kept that away. So if you have a smaller problem, maybe it's the thing to do.
Great talk, Danny. Some of this kind of goes to the idea of the microbiome in the built environment. In the same way we don't try to sterilize our patients because we find out that we do odd things when we try to squish as much as we can in one place. I mean, are we beginning to see some of that you're driving, changing the organisms that are there, and they do different things? They're good. They replicate fast. There's lots of them. So figure out how we kind of manage that microbiome.
Yes.
And again, you're coming up with some nice ideas.
Yeah, I think that's right. I think
So
our
do
hospital
you-
needs a fecal transplant.
So do you come up with a drain probiotic?
Yeah,
Is
that's
that
what
it?
Right. And there's somebody, and they, and so I've mentioned this, and of course, I mean, you can imagine, right? My hospital administrators are like, Mathers, no. Like, there's no way we're going to let you put bacteria down the drain that are then going to show up in a patient because of the risk issue, because it's just very difficult to study. So that is on our list of things to study. It's just tricky to study, like, when you get this ex situ model, and you've got your sync lab, and you're transporting, I mean, when you transport a trap, so we do that, we now have a BSL-2 plus lab where we transport traps from various hospitals or our own hospital into the sync lab, the microbiology immediately changes. And so there's a je ne sais quoi of the hospital, the things that are happening in the hospital that are really propagating this. And I think figuring some of that out is what we need to do. But I also think it makes it really hard to study things like what's the probiotic without getting somebody to agree to pour it down the drain. So anybody, we would be keen, but
biofouling is something that the industrial engineers have to deal with all the time.
Yep.
Cutting fluids and all sorts of stuff. And it's they, I don't want to say it, but in some cases they've just sort of given up. Because what you end up doing is you just end up kind of restarting and seeing how it goes. Start again, and just hope that you reach a good steady state of lower level biofouling, but it's never going to be gone.
that's right. And I think from a built environment standpoint, we need to think about, for me, I just didn't recognize all the wastewater sources in an ICU room, and I'm not sure they're all necessary. An ICU patient who is laying on a vent with a line is not getting up and brushing their teeth.
We're washing our hands, and again, this is I'm sure I've been to your hospital.
Yeah.
You've been to the other hospitals where good, I have to wash my hands in the patient's sink. And do I really want to be doing that for both?
Correct.
Or there's the sink down the hall. Okay, you don't want to go to that one.
So I think we just need to think about, we need to understand this so we can figure out what the right answer is. And adding data to that situation. One other thing that we know is some of the worst sinks, as you might, this is again all unpublished and a little bit of it's not 100% rigorous, but one thing that we have found is that the worst sinks are the ones that get used every 10 days or every, if it's used very, very frequently, there's municipal chlorine coming through that keeps things sort of that's speculation as to why it keeps things down. But the frequent use does keep the bacterial load in terms of quantitative bacteriology for KPC down. And so, if we had sinks outside of patient rooms, but frequently, but they weren't in that room, and then maybe not having, obviously, hoppers and toilets are probably a pretty bad idea to have in an ICU room. Hi, Dr. Love.
Thanks so much for the great I love the coming together of the different disciplines. I just want to offer that, and I don't know if this will be a good strategy for you, but we're considering this. One of the things we often do is we change it up on bacteria. The local barriers is one thing that's a key in from the water engineering field. We have to use local barriers to try to overcome them, but also change it up. So you keep one disinfectant over one thing, kind of PPC, they develop,
Right.
so you change it up periodically over time, and you get hard.
Yeah.
It's just,
I know. I memean, I think that's an interesting concept and potentially worth studying. What I would say is that with antibiotic, putting on my antibiotic stewardship hat, the data from cycling different antibiotic classes in ICUs at different times, use this empiric regimen for 3 months. So UVA did a lot of antibiotic cycling studies. We're actually the place where antibiotic cycling studies were mostly done. And it doesn't necessarily work. And it's Lou Rice that said, it's sort of akin to telling an alcoholic to drink wine for 4 months and vodka for 4 months and whiskey for 4 months and then beer for 4 months. it's all, you're all, combating. And so are you co-selecting for resistance when the mobile elements are so equipped to change it up? But, I don't know. Yes?
So let's somebody look at the shape of the thing, plasmid, and the length and all the flow from the process.
Yeah.
To see if it can limit inflation up.
Mm-hmm. So I've worked a lot with the UK. So there are a lot of countries that are farther ahead on this issue than the US. So in the UK, hospital sink, the bowl is designed completely differently. They would never have counter space adjacent to a sink. The outlet is the back of the sink as a hole in the back of the porcelain rather than a flat drain. They have sediment traps, which I wouldn't advocate for actually, but they have sediment traps rather than P-traps, which are slightly different. And faucets can never hit directly. Australian code is faucets can never hit directly. And the other thing that I've learned and Europe and Australia had already learned is get rid of overflows. There is no point in having an overflow in a hospital sink because it's just potential space. What is an overflow for? It is for if somebody left the water on, it prevents flooding, but it actually only prevents about 5 minutes before the flood hits because it usually doesn't have enough outflow. And in a hospital, there's not unattended sinks, especially in a patient room. So we shouldn't put overflows in. Code in Australia and Europe say no overflows. I advocate, we've gotten rid of overflows. I didn't even get into that, but we got rid of overflows in our surgical ICU and one of our thoracic cardiac units. Also, when they directly hit on the sink, the splatter is much more dramatic. So there's a lot of sink design, there's engineering solutions to some of these issues. We just need to get more recognition. And I think it is, that there are safer sinks and there are more dangerous sinks, that's what I'd say, or higher risk sinks for transmission, dispersion. Yes?
So what's wrong with your plasmid that takes it off plasmid?
So I don't know 100%, but it does not have an intact CRISPR-Cas system, is one thing. And so, CRISPR-Cas is great, it's a new technology, but it's really a bacterial immunity. And if you look at Pseudomonas that are multi-drug resistant, and you compare them to healthy Pseudomonas that are not multi-drug resistant, there's a significant difference between the CRISPR-Cas intact, drug-resistant Pseudomonas are less likely to have an intact CRISPR-Cas than antibiotic susceptible. And so I think that the sickly bacteria are picking this up and propagating it. Pseudomonas that also don't have a CRISPR-Cas have much bigger genomes on balance because they're just picking up stray DNA is one component of probably a really complicated situation of how plasmid,
So, Amy, if that's true, do you think, in fact, phage therapy would be better?
Maybe, but my environmental microbiologist that works with the PhD, he's a big fan, so we'll probably end up exploring some of that. I am slightly skeptical because I just think things are so complicated, and it's just the next change in this complicated biofilm that, and it's just one gene movement away into a different background, and so I'm not sure that it's the right solution, but it's a good idea and it should be tested. And in fact, the CDC is very keen on it, and so they're probably going to start looking at it as well.
So you mentioned Tara Palmer.
Yes.
So I know that she's learned more about plumbing than she ever wanted to know.
Yeah.
As well. But, and again, I know it's not published yet either, but I thought she found much lower rates of trap colonization than you did. And so what's different about either the patient populations or the municipal water or what? Have you guys compared notes to try to figure out why her levels seem to be a lot lower in the NIH Clinical Center
Yes.
than in their hospital?
We've compared a lot of, yes. So we have compared a lot of notes trying to get to the bottom of, because the concern to me early on was is there something with our watershed? that's unique, but I actually don't know how to completely understand the watershed of a region because we're not very, just to be clear, we're about 2.5-hour drive from the NIH Clinical Center. I think there's a couple of things. I think they caught their outbreak. I think our index patient seeded one of those drains in 2007. He was in a room where almost 50% of our acquisitions he was in a room in a unit in the surgical ICU where almost 50% of our acquisitions patients go through that unit. And so I think that we had a lot more time to develop a complicated biofilm before we thought about it. Whereas with the clinical center, they started ripping out pipes very early. And they ripped out to the wall. To be clear, they ripped out to the wall. And a lot of places have done this. I'm helping different hospitals. Millions and millions of pounds are being spent, or dollars are being spent, ripping out plumbing, which is not what I would advocate because I'm not sure that's going to work either. But they did that. We did not do that. We came to it and said, shoot, we've got this problem. Let's fix it. Let's study it. We shut down the unit, tried to understand it. And then when we changed out our pipes, a third of them were positive within 10 days again. And so is there something about the angle of our plumbing? I mean, I've spent a lot of time thinking, I work with plumbers a lot. And so, what's the angle of the drainage and what's the drop? And we have back-to-back sinks that actually probably aren't code in our surgical ICU where you can see through these back-to-back and there should be a drop. So maybe it's just reseeding from the common contaminated where we cannot get rid of it. So, I think it's a good question. And we've tried to figure out what's the difference. So, great. Yes, Les.
So would one solution be just getting rid of the traps entirely, if not for individual rooms, centralizing that sort of process somewhere that it can be more readily taken out and thoroughly sterilized on a basis?
Yes. Yeah. So there's no need for a P-trap except the sewer gas issue, which is a big issue. I mean, you're not going to have a happy patient if they have sewer gas smell in their room. But I think this is an engineering. I think I think the solution to some of this is in engineering. I think fighting it with chemicals, trying to figure it out, and all. I mean, with the complexity, I just don't think the solution is necessarily chemical warfare. I think it's engineering and built environment. And do we need P-traps? I don't think so. Do we need a sink in the ICU room, in a patient room? Maybe not. Maybe we do. Maybe we don't. We should study it. They did a waterless ICU in the Netherlands and had great outcomes. I mean, they're great outcomes. The outcome was they didn't have acquisition of drug resistance. Their rate of drug resistance across all species went down when they got rid of water in the ICU. And they did it, they committed. I mean, they did dry shampoo, only cloth bathing. It's a very nice paper by Joost Hoopman, actually. So, yeah, it's engineering, I think. Thank you guys so much.